Hello students, welcome to my educational channel Easy. In our last video, we've discussed about pteridophytes. Children, a few things slipped off my mind to tell you are regarding pteridophytes, which we will discuss right now, and then we will continue our next topic, that is gymnosperms. So, if you have not watched my earlier video of pteridophytes, please watch it, and there is some two or three points are in continuation of that video, that is pteridophytes. We studied that pteridophytes may be homosporous or heterosporous, means they can produce single kind of spores, or they can produce, some of them produce two kinds of spores, that is macrospores and microspores. If you remember, last video we have studied that macrospores or megaspores, they germinate to form female gametophyte, and microspores germinate to form male gametophyte. Now here the thing I want to tell you, that the female gametophyte in these plants, in case of heterosporous pteridophytes, the female gametophyte is retained on the parent sporophyte. If you remember, we have studied that the duration of the various cycles of plants, that is gametophytic stage or sporophytic stage, and their dependency varies. Okay, now what happens? The gametophyte, as we have studied, produces male and female organs. They have antheridia and archegonia. Antheridia produces gametes, that is male gametes, that is antherozoids. Archegonia, female, produces egg. Fertilization between antherozoids and egg takes place, and it results in the formation of zygote. Now all these things, see, they take place inside within the gametophyte. We have studied that gametophyte in pteridophytes is short, and the main part, dominant part of the life cycle is sporophyte. So female gametophyte in these plants, in heterosporous plants, is retained on the parent sporophyte for variable periods, means different plants have different periods for the retainment of their gametophytes. Okay, and the development of zygote into young embryo, that takes place in the female gametophytes. All right, fertilization takes place in the female gametophyte. Antherozoids reach the egg, zygote is formed, and this zygote develops into the young embryo, and this development of zygote into young embryo takes place in the female gametophytes. And just now I told you that female gametophytes are retained on the parent sporophytes, parent, not the new ones, parent sporophytes for variable periods. And this, as we have discussed it earlier, that this stage, that is the formation of embryo, this stage is the precursor to the seed habit, which is considered as an important step in evolution. And one more thing which I slipped off was that, see, seed habit, this is the precursor of seed habit. Zygote and zygote develops into embryo, which discussed it earlier. And one more thing which I slipped off, there was that a small classification of pteridophytes. The pteridophytes are further classified into 4 classes. And what are those classes? Only names you have to remember: Coelopsida, Lycopsida, Sphenopsida, and Pteropsida. These are the 4 classes of pteridophytes, and examples here are: Coelopsida includes Coelotum, Lycopsida includes Selaginella and Lycopodium, Sphenopsida includes Equisetum, and Pteropsida includes Dryopteris, Pteris, and Adiantum. Only this much I slipped off in my last video. So here it ends with the pteridophytes. Now today we will discuss about gymnosperms. Gymnosperms, that is the next group of plants. Till now we have studied algae, bryophytes, pteridophytes, and now gymnosperms. Now first we will discuss general characters of gymnosperms. See, gymnosperms include medium-sized trees or tall trees and shrubs. Right till now we have studied algae was thalloid, no differentiation of root, shoot and stem. Bryophytes, main part was thalloid, but somewhat root-like, stem-like and leaf-like structures were present. Then in pteridophytes we studied actual roots, stems and leaves were present. See the evolution taking place gradually, and so this is the sequence of their placement, and that's why I tell you every now and then you have to learn them all. Remember these names in hierarchy. Now comes gymnosperms, which include tall trees or medium-sized trees and shrubs. And you know, one of the gymnosperm, that is sequoia. One of the gymnosperm, the giant redwood tree, its common name is the giant redwood tree, that is sequoia, is one of the tallest tree species. See the evolution. Gymnosperm includes one tree, that is giant redwood tree, and that is one of the tallest tree species. Coming to the next character, the roots in the gymnosperms are generally tap roots. You might have studied in earlier classes types of roots. Now here full development has taken place, and now well defined roots and shoot system will be present. So roots are generally tap roots. Okay. Roots in some genera in gymnosperms. Okay. The roots in some genera have fungal association. Remember mycorrhiza we studied earlier also. So in some genera, roots have fungal association. Example, Pinus. Pinus is a gymnosperm. It is in association with fungus. Remember, symbiotic relationship, and that is known as mycorrhiza. Alright. In some cases, for example, cycas. See, cycas and pinus are the gymnosperms. In some cases, specialized small roots are present. These coralloid roots, these specialized small roots are called as coralloid roots. They are present in cycas. They are associated with nitrogen fixing bacteria, cyanobacteria, sorry. The roots of cycas, coralloid roots, those are specialized small roots. They are associated with nitrogen fixing cyanobacteria. Remember cyanobacteria? Blue-green algae. And so it helps in nitrogen fixation. Alright, we have studied this in algae also. Now, blue-green algae. Now, stem. As roots specified structures, characters, I have studied stems. Stems in gymnosperms may be branched or unbranched. Alright. Branched stems are present in Pinus and Cedrus. They are the examples. And unbranched stems are present in Cycas. So, roots, we have studied that they are mostly tap roots. And in some cases, they are in mutual association with fungus, that is known as mycorrhiza, and in some cases specialized coralloid roots are present which help in fixing nitrogen. Now coming to the stems may be branched or unbranched. Branched stems are present in pinus and cedrus, and unbranched stems are present in cycas. Now next, leaves. Leaves in gymnosperms may be simple or compound. Okay, these are the types of leaves. Leaves may be simple or compound. For example, in cycas, pinnate leaves persist for few years. In cycas, pinnate leaves persist for few years, and leaves in gymnosperms are well developed, well adapted. See, listen. In gymnosperms, these leaves are well adapted to withstand the extreme conditions of temperature, humidity, and wind. They have adaptation to survive in the adverse condition. How? See, in conifers, needle-like leaves are present. So what is the use of this needle-like leaves? Means leaves are reduced in the form of needle. You might be knowing the shape of the leaves. Alright, but here what you will see? You will see that in some cases you will find the leaves are simply needle like structures are present. All right, why? It reduces the normally the leaf is like this. Okay, there is more surface area, but when it is reduced to needle shape, what happens? The surface area is reduced. So in conifers, needle like leaves are present. And why? What is the advantage? They reduce the surface area, from the surface of the leaves evaporation takes place, water loss takes place. So what happens here in gymnosperms? To meet the extreme conditions of nature, temperature, humidity, etc., leaves are well adapted. So in conifers, needle like leaves are present, which reduce the surface area. And the leaves, they have thick cuticle. See here, there are some leaf adaptations. Remember it, they are all often asked in the questions in exams. So what are the various leaf adaptations in gymnosperms? Firstly, some of them are needle like. They reduce the surface area, and the leaf have thick cuticle. Cuticle means the outermost covering. The leaves have thick cuticle and sunken stomata. Stomata, you all know, there is the transfer of gases. Stomata are the structures present in the leaves. Sunken means they are not superficial, they are embedded, reduced. So in gymnosperms, in some plants, leaves have thick cuticle and sunken stomata, which helps to reduce the water loss. Both these adaptations are for reduction of water loss. In conifers, they are needle-like, and in some cases, they are thick cuticle. The leaves have thick cuticle and sunken stomata, which reduces the water loss. Now, next character. Gymnosperms are plants in which ovules are exposed. Exposed means they are not enclosed by any ovary wall. Ovules generally are covered by ovary wall, but in gymnosperms, ovules are exposed. That means they are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are also not covered. The seeds are naked. They are also not covered. Means ovule is not covered by any wall. It is exposed. Seeds are not covered. They are naked. And this is the name given to gymnosperm. What? Gymnos. Gymnos means naked. And sperma. Sperma means seeds. Plants in which the seeds are naked, that means the seeds are not covered by any wall, are called as gymnosperms. See, the hen's name is gymnosperms. It may be asked in exams. Alright, this is the specific character of gymnosperms, that ovules are exposed, seeds are naked, means they both are not covered by any covering. Now, next character, gymnosperms are heterosporous. We have studied in pteridophytes also. They may be homosporous or heterosporous. Now, here, gymnosperms are heterosporous, means you have studied that they produce two kinds of spores. What are those two kinds of spores? Microspores and macrospores. Alright? Micro means small and macro means big, larger. Two different kinds of spores are present. They are different in size. One is a small called as micro and other is large, big, that is called as macro. And both these spores, means microspores and macrospores, are n in number. That means haploid in number. Now, microspores, macrospores. All right. These are the two kinds of spores. And as we have studied till now, where are the spores formed? Spores are formed in sporangia. Sporangia is the spore bearing body. That means here, when we are heterosporous, when the plants are heterosporous, there will be different sporangia for microspores and different for macrospores. So there will be a microsporangia which will bear microspore, and there will be a macrosporangia which will bear macrospores. All right. Sporangia, that is the spore bearing body. We are studying since bryophytes. All right. Now these sporangia, where are they present? These sporangia are borne on sporophylls. Phyll, see, in biology, every word has its meaning. Phyll refers to the leaf. So, sporangia are borne on sporophylls, means some specific leaves of the plant which bear the sporangia, that is the spore bearing body, is known as sporophyll. Alright, and these sporophylls are arranged spirally on an axis. There is an axis on which these sporophylls are arranged spirally and they form a compact structure. Compact structure that is known as strobili or cone. Strobili or cone, or plural cones. Understand? Gymnosperm is heterosporous. It will produce two different kinds of spores, microspores and macrospores. Spores will be formed in spore bearing body, that is sporangia. Sporangia are arranged on specific structures, that is sporophylls. They are the leaf-like structures which bear sporangia. And these sporophylls are arranged spirally along an axis to form a compact structure which will be called as strobili or cone. Okay. So there are as two kinds of spores, micro and macro. They will be formed on different sporangium, that is microsporangium and megasporangium. They will be found on different sporophylls, microsporophyll and megasporophyll, and thus there will be two kinds of strobili or two kinds of cones, that is micro and macro, means male and female. Always we have studied that micro refers to the male and macro refers, or mega refers to the female. Now see, let's understand it properly, otherwise it will be confusing. So strobili or cones. These strobili or cones are present in gymnosperms which produce microsporophylls or megasporophylls. See, heterosporous condition is there. There will be cones of two types. One will be male and other will be female. Male will be called as microsporangiate. Male cone or male strobili is called as microsporangiate, and female cone or female strobili will be called as macro or megasporangiate. Okay, macro or mega. Now what will happen? These strobili or cones, male cone will bear or will bear what? Microsporophylls. Just now I told you that as it is heterosporous, sporangia and sporophyll will also be of two kinds, micro and macro. So the male cone bears microsporophylls and female cones bears megasporophylls. Alright, megasporophylls and microsporophylls. Strobili and cones are of two types, micro and macro, male and female. Microsporangiate will be a microsporophyll and megasporangiate will have what? Megasporophyll. Means microsporophylls are formed on the male cone and megasporophylls will be formed on the female cone. Okay. As a result, what will happen? Microsporophylls will bear microsporangia and megasporophyll will bear megasporangia. Again, sporangia, that is the spore bearing body. Alright. Consequently, microsporangia will produce microspores and megasporangia will produce what? Megaspores. Alright. See, male and female cone. Male cone called as microsporangiate or male strobili. The female called as the mega or macrosporangiate or the female strobili. Both the cones, male and female, micro and macro, will bear microsporophylls. Means microsporangiate will bear microsporophylls. Megasporangiate will bear megaspores. Sporophylls. In turn, sporophylls will bear sporangia. Microsporophylls will bear microsporangia and sporophyll will bear megasporangia. These sporangia in turn will produce spores, microspores and megaspores. Now these microspores develop into a male gametophyte. Alright? And female, that is megaspores, will develop into a female gametophyte. Now see, gymnosperms are heterosporous. Two kinds of spores are produced, micro and mega. Micro is male and mega is female. Now where are they formed? They are formed in the spore bearing body sporangia. Microspores on microsporangia, megaspores on megasporangia. Where these sporangia are present? They are present on the sporophylls. Microsporangia on microsporophylls, megasporangia on megasporophylls. Where these megasporophylls are present? They are present on their respective cones. Microsporophylls on the male cone or strobili, megasporophylls on the female cones and or strobili. And what they will form? Spores. What will they form? Microspore will form male gametophyte, will develop into male gametophyte, and megaspore will develop into female gametophyte. Understand? Now, this microspores which develop into the male gametophyte, we have studied that gametophyte. See now, gradually the phases are changing. Sporophyte is becoming the dominant phase of the life cycle. In pteridophytes also we studied that gametophytes are reduced. They are dependent on the sporophyte. Now here also the male gametophyte which is formed is highly reduced, okay, and is limited and is confined only to a limited number of cells, means male gametophyte is highly reduced and is made up of only limited number of cells. Now this male gametophyte is known as pollen grain. Okay, the male gametophyte in gymnosperms is highly reduced and is confined to only a limited number of cells, and this male gametophyte is known as pollen grain. Pollen grain, that is male. Okay, the development of this pollen grains, where will it take place? It takes place in microsporangia. It itself is a male gametophyte, but it is so much reduced that its development takes place where? In the microsporangia, where the spores are formed. Okay. Now, female gametophyte, that female megasporophylls forms the female gametophyte. The cones bearing the megasporophylls with ovules or megasporangia. The megasporangia we have studied here. Okay, as these megasporangia are formed on the megasporangiate, that is the female strobili. Now these male and female cones, and listen here, these male and female cones which I've studied here, listen carefully, they may be found on the same tree or they may be found on the different trees. Means in some cases, the male and female cone or strobili may be found on the same tree. For example, pinus. Remember the example. And in some cases, the male and female cones are found on the different trees. That is, example is cycas. Okay. In cycas, male and female cones are on different trees. Now, female gametophyte, which is developed from the female means megaspore, they bear archegonia. Okay, they see male gametophyte is reduced and is known as pollen grain, and female gametophyte bears archegonia. See, the megaspore mother cell, which is found in the megasporangiophore. Okay. The megaspore mother cell is differentiated. See here, I have explained it. This may sound somewhat difficult to you all, but understand it properly. See, the megaspore mother cell inside the megasporangia. Okay. It undergoes meiosis. It is divided by reduction division and it forms megaspores. And see how what happens in the megasporangia. Some cells are differentiated to form the megaspore mother cell. This megaspore mother cell is differentiated from one of the cells of the nucellus. Nucellus is the specific structure present in gymnosperm from which the megaspore mother cell is differentiated. So megaspore mother cell is differentiated from one of the cell of the nucellus. The nucellus is protected or covered by envelopes. By envelopes means by coverings. The nucellus is covered by coverings, that is envelopes, and this composite structure is called as ovule. Okay. And ovule, where it is born, the ovule is born on megasporophylls, which may be clustered in the form of female cones. All right. See, in the female cone, in the megasporangia, there is a nucellus. From one of the cell of the nucellus, megaspore mother cell is differentiated. Megaspore mother cell undergoes meiosis and form megaspores. All these things is taking place in sporangia. Now what happens? Nucellus, the specific structure is covered by some envelopes, some coverings, and they this composite structure is called as ovule. And where is it present? It is born on the megasporophyll. These megasporophyll may be clustered to form the female cones. Everything, all these things are taking place inside the female cone. So the megaspore mother cells divides by meiosis to form megaspores. And as you know, as a result of meiosis, how many megaspores will be formed? 4 megaspores will be formed. Of these 4 megaspores, you know, na, in meiosis, one cell forms 4 cells by the reduction division. Of these 4 cells, one of the megaspores, only one of the megaspores, here meiosis will take place and 4 megaspores will be formed, but only one of the megaspores enclosed in the megasporangium develops into a multicellular female gametophyte. See, microsporangia are present. Remember, just now we studied female cone will bear microsporophyll. It will bear microsporangia. See, everything is taking place inside this. Inside the microsporangia, there is a structure nucellus. One of the cell from the nucellus will differentiate into megaspore mother cell. This megaspore mother cell will undergo meiosis, reduction division, and form 4 megaspores. See, microsporangia will produce megaspores. Now this megaspores will be 4 in number. Out of these 4 megaspores, only one megaspore will be enclosed in the megasporangium. Only one will develop. Okay. Of the 4 spores, only one megaspore will develop into a multicellular female gametophyte. Alright. This one megaspore will develop to form the female gametophyte. And this female gametophyte will bear archegonia, that is the female sex organ. This female gametophyte, which is formed from the megaspore. Okay, one megaspore develops to form the female gametophyte. This female gametophyte is multicellular. It will bear female sex organ, that is known as archegonia, and they may be 2 or more in number. Means the female gametophyte will bear 2 or more female sex organs, that is archegonia. This multicellular female gametophyte is also retained in megasporangia. See, similarly, as pollen grain male gametophyte is reduced and its development it takes place in microsporangia. It develops in microsporangia. Similarly, the archegonia which are produced, this multicellular female gametophyte, see, male gametophyte reduced, female gametophyte multicellular. This multicellular female gametophyte will also be retained inside the megasporangium. See, the gametophytic phases are so much reduced that they develop inside the sporangium. Okay. Now these male and female gametophytes do not have independent free living existence. That is what I am trying to tell you, trying to make you understand that gametophyte phases are so reduced that they do not have independent free living and existence. They remain within their sporangia, retained on the parent sporophytes. That is what I am trying to make you understand by repeating it, that male gametophyte is reduced, it is developing in microsporangia. Female gametophyte, though it is multicellular, but it is also retained in its sporangia, that is megasporangia. And these sporangia are where? They are present on the main plant body, that is the sporophyte. See, so much dependency of gametophyte on sporophyte is here. If you remember all our plant groups, algae, bryophyte, pteridophyte, you will see that initially the main plant body was gametophyte, sporophyte was very much reduced, only limited to the formation spores. Then gradually there was a small phase of sporophyte. Then in pteridophytes we saw reduced gametophytes, a bit longer sporophyte, and the in pteridophytes also we have seen that gametophytes depend upon the sporophyte, but in bryophytes the thing is opposite. Sporophyte depends on gametophyte for its nutrition because that is green in color. So see the advancement taking place gradually. Now here male and female gametophytes are so much reduced that they do not have any independent free living existence, and so they remain in their sporangia, retained on their parent sporophytes. Okay, ovules are formed by the nucellus. Nucellus covered by envelopes borne on megasporophylls forms the ovule. Now what happens? See, I am trying to explain you with the help of the cycle. Okay, the pollen grain is released from the microsporangium. Okay, we have now here you have to study the reproduction or the life cycle. What happens? This male gametophyte is known as pollen grain. Okay, and female gametophyte bears the archegonia, that is the female sex organs. Now what happens? The pollen grain is released from the microsporangium. It is developing inside the microsporangia, so it will be released from the microsporangia. And this male gamete, that is what? Pollen grain, will travel to the what? Archegonium. Okay, how pollen grain is developing in microsporangia, it will be released from the microsporangia and it will travel here, where? Up to the ovule. And where is this ovule? Ovule is inside the megasporangia. Okay, and how it will travel? It will travel with the air currents. So pollen grains will be reduced from the microsporangium and are carried by the air currents and come in contact with the ovule, where at the opening of the ovule born on megasporophylls. Just now I told you that ovule are present on the megasporophylls. Okay, see, megasporophylls, ovules are present. Male gametophyte is released from the microsporangium and it will travel to the opening of the ovule, where it is present, where ovule is present. Ovule is present on the megasporophylls. All right, this will travel to the opening of ovule born on megasporophylls. Now what will happen? Pollen grain will form a pollen tube. This pollen tube will enter the, it will grow towards the archegonia. It will end archegonia present in the female gametophyte. Okay, sporangia. Now pollen tube will be formed growing towards the archegonia, and this pollen tube will carry the male gametes towards the archegonia in the ovules and discharge those contents near the mouth of the archegonia. Pollen grain will be released from microsporangia, will be carried through air to the megasporophyll where archegonia is present. Ovule is born on the megasporophyll. A pollen tube will be formed. This on the mouth of the ovule. This pollen tube will carry the pollen male gametes where to the mouth of the archegonia and will discharge the contents near the mouth of archegonia. As a result, fertilization will takes place. Now see the cycle, life cycle, reproduction. How does it takes place? Sporophyte, main plant body is sporophyte. It bears, it is heterosporous. It bears microsporangium as well as megasporangium. Maybe in some cases they may be on the same plant and in some places they may be on the different plants. The main plant body, sporophyte, will bear microsporangium and megasporangium, male and female. Microsporangium will produce, just now we discussed, I told you, no need to revise that they are formed on microsporophylls and megasporophylls, which form the cones or strobili. Okay. Now microsporangium will produce microspores, that is male spores, and megasporangium will produce megaspores, that is female spores. Both the microspores will develop to form male gametophyte. Mega or female spores will develop to form the female gametophyte. Both these gametophytes are reduced and they are developing. They are retained in their sporangium. They are reduced, retained in the sporangium. This male gametophyte is known as pollen grain and female gametophyte is multicellular and will produce two or more archegonium inside the archegonium. Within the sporangium with megasporophyll, ovules are present. Now what will happen? Pollen grain will be released from the microsporangiophore and it will travel through air currents to the mouth of the archegonium. A pollen tube will be formed from to the and releasing the male gametes to the mouth of the archegonium, and now here the fertilization will take place. Pollen grain will fertilize with pollen grain will fertilize with the ovules. It will reach the ovules. Okay, it will reach the ovules inside the archegonium. Fertilization will take place. As a result, zygote will be formed. As a result of fertilization, zygote will be formed. Now this zygote will develop into embryo. Similarly, as we have studied earlier in pteridophytes also, zygote will develop to form embryo, and this embryo will now develop to form sporophyte, a new plant, new sporophyte. And here one thing, that zygote develops into an embryo and ovules which were present inside the archegonium, where the on the mouth of the ovules pollen grain has released the male gametes. Okay, those ovules, they will form seed. Zygote will develop to form embryo and ovules will develop to form seeds. Remember it? Here the seeds have started. In pteridophytes we have studied zygote develops into embryo and this is the precursor of seed formation. We have studied in pteridophytes. Now here we have specific ovules which develop into seeds. Okay. See, similarly, gametophyte condition, that is diploid condition, is main. Sporophyte diploid, sporangium, spores haploid, gametophyte haploid, pollen gametes haploid. Fertilization takes place. Zygote diploid, Embryo diploid develops into new sporophyte, that is diploid. Dominant phase of life cycle is diploid, that is sporophyte. Embryo develops to form zygote develops to form embryo, embryo develops to form sporophyte, and ovules develop to form seeds. Here a new character has started, seed formation has taken place. So this was all about gymnosperms. Please like, share and subscribe my channel. Thank you.